Okay, good evening everyone. Before going to start embryology, I would like to mention few things with regards to how you have to prepare for any examination. Actually, I tell you the mode of preparation that I have done when I am student like you. The first thing is goal setting. So I have to set a goal for myself. So I wanted to do my MSc only in Andhra University. So that is my goal. So I did not write for any other university. So my intention is to get into Andhra University. That is goal. So your intention should be some other university or central university. Second thing is what subject you want to get into. So my aim is to become a zoologist. I got 90 plus score in mathematics. Still, I got into BPC in intermediate. Though I got 90 in 10th class. Why? Because I prefer only geology, natural sciences to that of mathematical. Though I am proficient in that aspect, in that subject, my orientation is towards this. That's why I've taken this. So, second thing is your goal should be what subject you want to pursue in MSc and what your intention of doing MSc in geology or botany or chemistry. So, what actually my intention is that I want to get all the knowledge with respect to geology up to what we call 10th class. We have all subjects: social, maths, languages, science, etc. After 10th class, when we get into intermediate, social component is over because we are science students. And we have maths, physics, biology, botany, chemistry, in addition to languages. By the time we come to degree, physics is gone, chemistry, botany, zoology. And now all are gone, only single major subject. And by the time we get into MSc, only one subject. So that subject, because you're studying one subject for the entire 2 years, you will get more proficiency. That means more knowledge in that particular subject. That's it. And one can have another option that after completing my MSc in zoology, I want to become a zoology lecturer. I want to become an IFS officer where there is a optional for zoology as well as botany. There is an UPSC examination for civil services where there is an optional for zoology because I have completed MSc with lot of knowledge in zoology. I can score much. So these should be the next criteria for you. And after that, you have set your goal, you have selected your subject, and what your intention by taking up that subject is. Now we have to start preparing. So what I have done is I wanted to become a civil servant. So what I have done is first I wanted to know what actually the paper mode of question paper is. So I have seen that there is a preliminary, there is a mains and interview. In the prelims, there is general studies component, just general studies component. But actually when we were writing, there is general studies and one optional, and I wanted to choose and score more in that optional, that is zoology. That's why I went for MSc. For optional in civil services, when I was writing that, there are 120 bits and each bit was 2.5 marks. And for general studies, there are 150 bits and each scores only 1 mark. That means if I get more marks in my subject, even if I get less marks in general studies, I can clear prelims. That is the first thing that you have to know is the examination pattern. The next fifth thing that you have to know is what actually in zoology or general studies is asking about. So I have taken a small booklet in the market where it is UPSC zoology optional last 10 years previous question papers without solutions I've taken. Likewise, general studies previous 10 years questions. Now I take the zoology paper. I see the first question from where it actually is. So I take and write there. This first question is from cell biology. Second question, again cell biology. Third question, it is from invertebrates. Invertebrates in brackets protozoa. If it is from protozoa. And fourth questions, it is from chordata. In brackets again cyclostome or fishes, whatever it may be. So likewise, I analyze for the entire paper for 1 year and write down how many questions are being asked from cell biology, how many are from genetics, and how many are from embryology, physiology, invertebrates, and that too in invertebrates, which phyla are going to be asked from which phyla more questions are going to come. After analyzing, I cannot fix myself to this particular analysis because that is for only 1 year. Next, I go for the second year, third year, fourth year, fifth year. So, whatever the previous papers available to me, whether it is 1 year, 2 years, or 3 years, or 4 years, or 5 years, or 10 years, I go through the entire content, and this takes me this what we call takes a time of nearly 1 week for me to analyze it. So after that, I put all the papers. For example, this is 2024. So 2023, 22, 21, 20. I take a big sheet, one tall sheet, and above that I write invertebrates, vertebrates, and so on. So and how many questions from 2023? How many from each section 2022? I note it down and try to conclude from which part more number of questions are coming each time. And now my mode of preparation will be to take that subject. Okay, for example, about 15 questions are coming from physiology. I'll start preparing from physiology component first, followed by the next, followed by the next, and followed by next. Don't think that embryology is said to be from embryology only two questions are asked, but there are so many topics. So I need not study. That should not be your spirit. You have to score more in each subject, in each in each content, isn't it? So what you have to do is start preparing with those that are from which more questions are coming. Then go to the other components and try to finish everything. So for this, you have to start your preparation from the third year itself. That's the reason why even while you are in sixth semester itself, I have started this coaching. Okay, so this should be your mode of preparation. When you prepare like this, sure shot any subject, if you're going for a group 2 examination, group 1 examination, or any SPA clerical examination, SSC, anything, if this is your pattern of study, then you're going to get that job sure shot. So after analyzing, what you have to do is you have to start preparing, for which you have to collect books. So in these days, you need not have to collect any book. So much so material is there in the internet. So previously what we are doing, we are taking the question papers. There are solved ones and without solutions also. We take the without solutions paper so that we can know, we will be answering, and after that we check with the solutions booklet, and from there we come to know that so which section I am strong and which section I am said to be poor, so that I can consolidate that section also. That should be the last step. If you prepare like this, you will succeed in your life. So don't ask me. You do it by yourself. You'll be knowing what you have to do. If there are more questions from biochemistry. Biochemistry is not your cup of tea in your degree. Then you have to start preparing for biochemistry because you haven't studied that topic in your entire degree for just a semester. That too, only one unit you have studied. So change your preparation. Before that, analyze the question paper, then you will get what you want. Okay. So with this brief introduction of how to prepare for the examination, I will now move to the embryology component. Coming to embryology, previously when I was studying, this is called as embryology. Now this is being called as developmental biology. We don't use the term embryology now. Etymologically, it is derived from a Greek word, embryon, which means the unborn, and in English we call it as embryo. And logos, we know it is study. Thus, it deals with the study of the embryo, simply speaking, embryology. But actually, we are not studying the embryo here. We are studying the process of the development of an individual, all the biological aspects involved in the development of individual, starting from zygote till the adult is formed. Hence, we rather call this subject or branch of science as developmental biology instead of embryology. So it includes the prenatal development of gametes, that means starting from gametes, followed by fertilization and the development of the embryo are the aspects that are covered in embryology, which we now call it as developmental biology. When you go to MSc, you should not study the general books of embryology. There is a book by embryology written by Verma and Agarwal. It's a very nice book. You can go for it. But in order to understand what actually the developmental biological aspects in detail, you have to go and refer in university two books. One is Embryology or Developmental Biology by Balinsky. The other is Developmental Biology by Scott F. Gilbert. So these are the two main books which we have studied while we are in MSc Geology, in an MSc, and these are the two important reference books. Along with this, there is Embryology by Rastogi, Embryology by Verma and Agarwal. These are the main books. You can get them in PDF form also from internet. I would like to start with certain terms associated with embryology first. The first term is said to be development. Development is the course by which a complete multicellular organism arises. So the complete multicellular organism can arise from a fertilized egg or an unfertilized egg. So if it is arising from a fertilized egg, the means is sexual reproduction. If it is arising from unfertilized egg, the means is a parthenogenetic mode of reproduction. Whatever it may be, we are studying the development of a complete organism from an egg, whether it be fertilized or unfertilized. That is what development is. And coming to the next one, embryogenesis. It is the development of a new individual from a fertilized egg or zygote, where we study or focus mostly on the development of embryo. So both are seem to be the same, but when we see development, it involves all the biological aspects involved in the course of this entire embryogenetic genetic process. The next one is blastogenesis. Blastogenesis is the development of an individual from an asexual reproductive body. There is sponges. Sponges are sedentary and they give rise to another sponge by budding. So by budding, which is called as external budding, body fragmentation during regeneration. In case of sponges again, gemmules. Gemmules are internal buds from which sponges can develop. All these come under what we call it as blastogenesis. Next one is epigenesis. It is a gradual increase in organization and complexity from simple initial structures. So when we are in embryonic form, we do not have all the complex structures that are present in an adult. All these develop from simple structures, and this process we call it as epigenesis. Next one is morphogenesis. Morpho means form. Okay, form means shape like thing. Genesis means development or origin. So the creation of form is called as morphogenesis. That means the growth and division of cells, interaction of these cells with the other cells, with the extracellular matrix, and eventual death of certain cells is what we call it as morphogenesis. That leads to the development of the individual. So the shape of the previously this is a ball-like structure, the embryo. After that, it becomes a fetus. After that, we develop all organs and organ systems, and we got a particular form. That is what we call it as morphogenesis. Okay. Next, cytogenesis. Differentiation of undifferentiated cells of the germ layers into specialized or functional cells of the body is called as histogenesis, and it leads to the next process, that is organogenesis. Here we have an embryo with outer ectoderm, inner endoderm, and middle mesoderm, three layers. Hence we are called as triploblastic animals. And these are called as germ layers or germinal layers, because from these only all the different parts of the organs of the body germinate. Ectoderm gives rise to the simply speaking body wall and also nervous system. That means there are certain cells here, all are ectodermal cells only. That means they are not distinguished to form nervous system or skin cells. They are undifferentiated. But during the course of development, some of the cells form into nerve cells, some of them form into body wall, skin cells. And this process we call it as differentiation. So this differentiation of undifferentiated cells from germinal layers to specialized cells of the body is called as histogenesis, which leads to the formation of organs called as organogenesis. And coming to the next part of the embryology, this is introduction only, that is theories. Aristotle is called as the founder of embryology. We can also call him as the father of embryology because he is the one who told us about animals, hence he is called as father of zoology. And he told us about animals and plants, hence he is called as father of biology. And he also told some aspects of embryology, embryonic development, hence we call him as founder of embryology or father of embryology. And coming to what he has done, he wrote a book that is called as Generation of Animals in English, and in its Greek language, De Generatione Animatium. It describes the reproductive aspects and the development aspects of many different types of animals, and hence we call him as this thing, founder of embryology. He also coined the term epigenesis. Okay, what epigenesis is we'll know later. and he coined another term called as entelechy. What is entelechy? He called soul, we call it as Atma, the non-material purposive principle that guides development. So he combines physically appearing embryo with that which is not clearly seen by any organism, that is the inner soul. So according to him, the entire development is guided by the soul, and this principle is called by him as entelechy. Next, another theory is the theory or any dictum proposed by William Harvey. You know about William Harvey, about circulation. He is famous. He wrote another book, Exercitations de Generation Animalium. And here in this book, though he mentioned animals, it particularly deals with the development of only chick. And after studying this, he proposed a dictum. This called as ex ovo omnia. Ex ovo omnia. That means all animals arise from eggs only. That is what he discovered. Next one is preformation theory. And this preformation theory is proposed by a scientist called as Jan Swammerdam. Along with him, Marcello Malpighi also advocated this theory. But if there is a question on who proposed this, A. Jan Swammerdam, B. Marcello Malpighi, C. somebody else, D. none of the above, then you have to choose between these two, and then you have to choose only Jan Swammerdam. Though both advocated the same, but this is Jan Swammerdam who proposed the theory, which states that sex cells contained preformed embryos or miniatures of the adult. According to them, there is a sperm. Inside this sperm, if this belongs to a bird, the bird's preformed bird is present within the sperm. If this belongs to a reptile, the preformed reptile is present within the sperm. If this sperm is belonging to a human being, here the preformed human being is present within the gamete. Thus, sex cells, whether it be sperms or ova, according to this theory, contained the preformed embryos, which are nothing but the miniatures of the adult. So, upon fertilization, this preformed embryo gives rise to the organism. And based on this, a scientist called as Hartsocker, he published a drawing in a particular journal called as Des Scavens in 1695, where he has shown a miniature of the adult human being within the head portion of the spermatozoon, and he called it as homunculus, means little man. The next one is that after preformation theory, they are saying that sex cell contained preformed embryos. That is not mentioned whether it is sperm or ovum that is responsible. Though William Harvey said that all animals are arising from eggs, there is a divide between the scientists. Some are called as spermists and some are called as ovists. Ovists believe that ovum contained the miniature adult. Spermists believe that the head of the sperm contained the miniature organism. Based on this, some say that it is the ovum that gives rise to the adult. Some say that it is the sperm that is responsible for the adult. Respectively, they are called as ovis and spermis or animalcules. Leeuwenhoek and Hartsocker are spermists. Marcello Malpighi, Jan Swammerdam, Haller, and Bennett are called as ovis. And next one is epigenetic theory. It is proposed by Caspar Friedrich Wolff. This is one of the most important theories you have to remember. epigenetic theory is proposed by Caspar Frederick Wolff. According to this, organs are formed successively in epigenetic manner. So, what is this epigenetic manner? The granules of globules of egg. That means, according to him, the egg contains granules. They organize into rudiments. From these, the rudiments of germ layers arise, which develop into various organs of the embryo, whether it be liver, whether it be heart, whether it be kidney. All these develop from the granules of globules of embryo, according to the theory proposed by Wolff. Next, most famous theory is recapitulation theory. It is also called as biogenetic law. It is proposed by Muller and Ernst Haeckel. According to this, ontogeny repeats phylogeny, simply speaking. Because it is called as recapitulation, means repeating, we can call it as ontogeny is a recapitulation of phylogeny. What does it mean? Ontogeny is the history of an individual. Where from the history of an individual starts? From the formation of gametes, followed by fertilization and development of zygote, which leads to the formation of adult and till his death. This is what an individual's history is. Leave about this death. And by the time we are born, we have all the structures that are present in the adult. So up to the formation of the adult baby. That is what an individual's history is. What the scientists say the individual history repeats his or her ancestral history. The history of an individual repeats his or her ancestral history. Scientists say that just before birth we lose tail. That means we have an actually tail while we are as a fetus in the mother's womb. What does it refer to? It refers that our ancestors are reptiles. And scientists say that before lungs are formed, we have gills. That means our ancestors also include fishes. So, thus, according to recapitulation theory, our history repeats or shows the ancestral history. And another theory is called as germ layer theory, proposed by von Baer. And he is called as the father of modern embryology. Aristotle is the founder of embryology, and for modern embryology, the father is von Baer. According to this, various structures of the body, that means all the structures, all the body organs, arise from germs layers in different species of animals from the same germ layers. That means if ectoderm gives rise to body wall and nervous system in human beings, it give rise the same to other mammals, other reptiles, other birds, other fishes, other amphibians too. So that means from the same germ layers in different species of animals, all the body organs or structures arise according to the germ layer theory. Later, von Baer proposed a theory that is popularly called as Baer's law. This is called as Baer's law. According to this, in the embryo, more general features that are common to all the members of a group of animals are developed first, followed by special features which distinguish the various members of the group. For example, we are primates. Okay, so primates are distinguished with certain characteristics. We have a bipedal gait. We walk on two legs, isn't it? But when we were born, we are almost crawling as if we are having four legs. It suggests that we are tetrapods. Okay. So first of all, what happens? General features that are common to all the members of groups of animals. So what is this group here? Tetrapod, from amphibia to mammals. So all the features that are common to all these group of animals develop first, followed by the specialized features. For example, amphibians. What is their specialization? They can live in land and they can live in water, they can live on land, for which they have buccal respiration, they have pulmonary respiration, and also they have uh, what we call aquatic respiration in larval form. And in addition to this, there is what we call uh, cuttlefish respiration. So these distinguish amphibians from others. And what about the birds? Birds, when we speak of birds, we say feathers. So these are the special features that only birds have. And coming to the primates, mammals, like human beings, we have hair on the skin. So this distinguishes us from the other animals of this group, that is tetrapod. Thus, the general features are formed first, which are common to all the animals of a group. And as the embryonic development proceeds, special features are said to be replacing the or said to be added to the more common features. And this theory is said to be a big blow to recapitulation theory. Thus, according to Bayes' law, or what Bayes suggests is, ontogeny does not repeat phylogeny. Why? An embryo does not pass through the adult stages observed in animals. According to him, the adult stages observed in animals, that means we are said to be having an ancestors, reptiles, amphibians, before that fishes. If that is the case, we should have the adult amphibian characters or adult reptilian characters and adult fish characters according to recapitulation theory. But actually, we are not having those characters according to our Bayes' law. What he says is early embryos share common characteristics and become more and more divergent. That means they are specialized as development proceeds. And this is a path-breaking find in the science of embryology. Hence, we call him as the father of modern embryology. And let me also tell you some other terms associated with embryology and who coined those words. For the first time, a scientist called as Steno introduced the word ovary. R. D. Graff discovered follicles in human ovary. That is the reason why, based on him, the mature follicle is called as Graffian follicle. Why we call it as Graffian follicle? Because this name is, or these are discovered by Graff. And Anton Leeuwenhoek observed sperms under microscope and he called them as animalcules, smallest animals, little animals according to him. And Von Baer discovered the mammalian ovum for the first time in 1827. Wo Hetwig, you have when if you have attended the cell cell biology classes by Binduna, you have heard his name, Wo Hetwig. There are cell theory, organismal theory, and protoplasmic theory. Organismal theory is said to be proposed by Hertwig. So this O. Hertwig concluded that fertilization involved the union of sperm and egg nucleus. So for the first time, the concept of fertilization is elucidated by Hertwig. Okay, and another scientist H. Fall, by going a step ahead, He observed the penetration of sperm into the ovum. How the sperm penetrates into the ovum is observed by him. And another scientist, Charles Otis Whitman, initiated the study of cell lineage by tracing the detailed history of the cells from the dividing egg in a leech scientifically called as Clespin. So what is cell lineage? Here there are cells in ectoderm, isn't it? So this cell gives rise to body wall, isn't it? And these cells give rise to nervous system. And some of these cells which give rise to nervous system, they also, so they are differentiated to form cells which can distinguish into nervous tissue. And from there, are going to develop brain and some are going to develop into spinal cord. And after that, cranial nerves, after that, spinal nerves, and so on. That means this is the what we call uh, history of this particular cell which differentiates to form different kinds of cells. And this is what we call this as the ancestry of a cell, ancestry of an organ starting from a particular cell. We call this as cell lineage. This concept is developed by Charles Otis Whitman. And Jacques Loeb is another scientist who discovered the phenomenon of artificial parthenogenesis. And coming to lastly, in embryo, while we study embryo, we also see that certain children are born with malformations, okay, with legs undeveloped, with brain undeveloped, with hands undeveloped, and so many complications. And these complications or malformations that are seen by birth are called as congenital malformation. And for this, there is a Greek word called as terata. And thus evolved another branch called as teratogenesis, another term called as teratogenesis. Teratogenesis is the study of congenital malformations. That means that malformations that are seen from birth. And while this is being done, scientists found that for the mother, okay, during operation, they give certain sedatives, anesthesia we call it as. And for this, they used to give a tranquilizer, a sedative, and that is called as thalidomide. So before, most probably about 30-40 years back, thalidomide is the tranquilizer that is used by the doctors. And later it was found that those mothers which were tranquilized by thalidomide, which were tranquilized with thalidomide, are giving birth to what we call babies that do not have limbs, a condition called as ectromelia, and those babies are called as thalidomide babies. So after the discovery, they started what we call banning this particular tranquilizer. And another two terms that we can see with respect to congenital malformations are spina bifida. That means spinal cord protrudes out. And another one is anencephaly. Cephala means head and means something, a defect or absence. Defect in the development of head and brain is called as anencephaly. So these are the different terms that I wanted to discuss with you. Now we'll start with the actual embryology with gametogenesis. Before going to that, I would like to mention few aspects of the embryonic development so that tomorrow we'll discuss the gametogenesis, fertilization, types of eggs, types of egg membranes, process of cleavage, and gastrulation. Up to this, there is a syllabus in your degree that I'm going to deal with tomorrow. And today we'll discuss about actually what is happening during the embryonic development. Before that, I would like to ask you one question. When you write general characters, okay, general characters of protozoa to chordata, That is the one of the important questions while you are intermediate as well as degree. And the last point in general character is you write that development is direct, or in some you write development is indirect. What do you mean by direct development and indirect development? Can anybody tell me?
So direct development
Having
means
larval form is indirect,
direct
sir.
give to birth
Without
baby cells.
larval form is direct.
Indirect development means there is larval forms.
Very good, excellent. So when there is a larval form, that is indirect development. So fertilized egg followed by larva followed by adult. Fertilized egg, if directly gives rise to adult, we call it as direct development. Why certain organisms? Next question is why certain organisms are having indirect development while the others are having direct development? What could be the difference? Why the animals having direct development are not showing indirect development? Likewise, why the animals showing indirect development? Why they are showing only this? Why not direct development? Is there any answer?
Those eggs may be different, sir. Like for indirect development, the embryo, for developing that, they require the food. For indirect development, there is no sufficient food inside, sir.
Very good. One
So
answer is
direct indirect development there is a placenta is present.
so for indirect, for direct or indirect.
For direct development, placenta is present. The nourishment is taken through the placenta, sir.
Okay. Then what about the development of a chick from egg? Do you call it
Yolk
as
sac plays an important role to nourish the embryo.
so here there is no placenta, only yolk sac is there. Then you call it as direct or indirect development.
So it is indirect development. No, 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 sir. It is direct
Indirect,
development.
yes.
It is direct development only. So need not be placenta. Is there any other reason other than food? Why actually a particular organism is having a larval stage? Why you should have a larval stage? Why not an adult come? Larva is not bigger than the adult. Larva is not mature than the adult. It is somewhat lower graded animal than the adult, isn't it? Then why these animals are choosing larval form in their embryonic development or in their life history? Who is going to respond? Hmm?
If it is in one place, there will be more competition, sir. If it moved to some other place, uh, we consider food or mating or not mating. After development, mating and shelter also, it will be made.
Very good. Excellent. So there are two aspects here. Both were told by you, and for the understanding of the others, I'm dealing with one once again. The first aspect is nutrition. So during the development of male gametes, all the yolk material, sorry, all the cytoplasmic material is lost. Very less cytoplasm is present. Why? Because this is the one which has to move towards the egg. So in order to enhance motility, it will not accumulate food. And the one gamete that accumulates food is the ovum. That is why it is larger than the sperm, and it accumulates all the food material, and thus it is stationary. It is not motile. Only the male gamete or the sperm searches for it. The second thing is once the sperm reaches it, it only transfers its nucleus and then it falls off. What happens now? The entire development is taking place inside the egg, which is called as the fertilized egg or zygote. Thus, This zygote should contain all the food material that is required for the embryonic development. There are certain types of eggs which are called as microlecithal eggs, and certain are called as macrolecithal or mesolecithal eggs and mesolecithal eggs. Microlecithal eggs have only very little amount of yolk. Macrolecithal eggs have large amounts of yolk, even mesolecithal also. And when this is the case, what happens is micro, meso, and macrolecithal eggs. In case of organisms where there are microlecithal eggs, the food is sufficient only from zygote up to the early embryonic stage. Then what about the remaining part? It cannot develop because there is no energy. And for energy, nutrition is required. And those nutrients are exhausted by the time it becomes an early embryo. Now, the organism has to develop into adult and it requires a stage which can procure the food material for which it has to move. So, thus, this develops into a larval form. The larval form has two characteristics. One, that it can move, the other, it can feed. These are the two characteristics of a larval form. It cannot develop into it cannot reproduce. Why? Because it is sexually immature. This is another characteristic. So, a sexually immature larval form developed from a microlecithal egg or even certain organisms where there is macrolecithal egg. It moves, collects feed, and completes remaining embryonic development and becomes the adult individual. So those organisms where there are microlecithal eggs and mesolecithal eggs, they need a larval form. Okay. And the second aspect is that about the macrolecithal eggs. They have enough food material in the egg so that within the egg it can complete its complete development and come out as an adult. And hence we call this as a direct development. So direct development is related to the amount of yolk that is present within an egg. Okay. And coming to the human beings have a microlecithal egg. So we should have a larval form in our development. But still too we are not having larval form. Why? Because as someone said, Lakshmi or someone, we have a placenta and the entire embryonic development during this period, the placenta feeds us. So this is the first aspect with respect to what we call development, which is direct or indirect. The second one is said to be the stationary habitat of the organism. For example, there is a sponge. Sponges are said to be stationary, or we can call them as sedentary animals. That means they cannot move. If they cannot move, what happens? They release their zygotes or sperms or ova into the water where fertilization takes place and zygotes are produced. And the zygotes develop into adults. If all the sponges that are present, about 1 lakh sponges are present in a particular area, and each one gives rise to thousands of eggs, that means 1,000 lakhs of sponges will be formed. At the same place, if that is the case, there is deficiency for water, there is competition for what we call oxygen, food, and other aspects. In order to avoid this, those organisms which are sedentary are also having microlecithal eggs. So that what happens? They develop into an larval form which is responsible for what we call it as dispersal. Because it can move in search of food, it goes away from that place, and wherever it has food material sufficient, there it gets onto a substratum and develops into a adult, which becomes a sedentary animal. So these are the two aspects. One is nutritional aspect, the other is the sedentary habitat of the animals. That is what is determining the type of development. And let me start with what actually happens here. So here there is a sperm and ovum being formed, and they undergo, sorry, just a second. So sperm and ovum, they undergo fertilization to produce a zygote. Okay, so zygote is formed. Now this zygote starts undergoing cleavage because it is unicellular and it has to produce a multicellular adult, for which it has to undergo divisions. All the divisions that take place during cleavage are mitotic divisions only. Now it goes on dividing and forms into a group of cells which are in the form of a mulberry fruit to the scientist who first saw them, and hence he coined the term morilla for this. So morilla is based on the shape of the what we call this embryo, which resembles that of a mulberry fruit. And here the cells are arranged. These are not specifically arranged, they are said to be randomly arranged. They are into ball-like structure. What happens? There are so many theories which we'll discuss later. One theory states that water comes out of this each cell into the interstitial space between the cells, so that gradually this moves all the cells that are present inside towards the periphery, so that what happens is it organizes into a structure where the cells are present outside, leaving a cavity inside, and this is called by them as blastula, and the cavity is called as blastocoele, and the outer layer is called as blastoderm. This is single layered, and this blastoderm is called by scientists as ectoderm. And the process of formation of blastula is called as blastulation. So now we have discussed about two processes. One is morulation followed by fertilization. Second one is blastulation, that is the formation of a blastula. Now what happens is all these events take place inside the egg only. So this is the egg with its nucleus. The egg is surrounded by another membrane called as vitelline membrane, around which there are secondary membranes and tertiary egg membranes. So whatever the cell is going to divide, it is confined to the interior of this much space only, isn't it? And in this space only, what is happening? The blastula is said to be formed with cells towards the periphery. Now what is happening? Each cell is again dividing into two. So from these two cells develop, from these two cells develop, from this. But in order to, if the cell number increases, it should increase in size. But the space is said to be limited. So what happens is, instead of increasing in size, there is a gap inside. So some of the cells which are increasing in number due to division, they go inside. They go inside, forming into another layer. So thus, from blastula another embryonic structure which is having cells that are outside. Due to division, as the number of cells increases, they go inside the blastocele and form into a two-layered structure which we call it as gastrula. Okay, so this gastrula is two-layered. Previously it is only one layered blastula, and now it is two layered, and the outer layer is again given the same name, blastoderm or ectoderm. Now we prefer the term ectoderm, and there is another layer called as endoderm. Okay, before explaining what happens afterwards, let me explain the structure of the human body or any other organism. So what happens here is the human body, when we take up the structure, okay, we have, starting from mouth till the anus, there is an alimentary canal at the center. We have alimentary canal at the center, and this alimentary canal is present in us on the ventral side. This is our organization. And so if we show this organism like this, so this is the alimentary canal. Okay. This is mouth, alimentary canal, and anus. And around this, there is a body wall. There is a body wall around the alimentary canal. And in between these two, there is a body cavity. Isn't it? There is a body cavity. And before that, on the dorsal side, we have a notochord, above which there is a nerve cord. And inside the body cavity, we have all the organelles. That is heart at this place, lungs here, and kidneys here, and all the remaining parts of the organs of the body. Now, all these different parts with different cells are derived from the cells that are present either in the ectoderm or in the endoderm. That means some of these cells have to develop into skin, that is body wall. Some of these have to develop into alimentary canal. Some of them have to develop into heart, kidney, so, and also body cavity, including in addition to nervous system, that is notochord and also nerve cord. So now what happens is, in the case of poriferans and coelentrates, what happens is the embryonic development is up to this point where two-layered gastrula is formed. From there, the adult develops after completion of embryonic development. Hence, we call them as diploblastic animals, where all the different organs or the parts of the body are developed from only two layers of the embryonic development. They germinate from these two layers. Hence, these are called as germinal layers, and they are two in number, and hence they are called as diploblastic animals. Whereas in other organisms, that is not the case. Some of the cells which are present at the junction, some of the cells which are present at the junction between the ectoderm and endoderm, these are the cells. So, as the embryo grows, blastula undergoes division, more number of cells are produced, certain cells go into the interior, form into endoderm. And still the same, the cell division continues, and there is no space for enlargement of the developing embryo, but there is a space here where the cells can go. And now these cells, that is the cells which are present at the junction of the ectoderm and endoderm, they start migrating inside. They grow inside. From both the sides, they go inside and they form into a third layer. And this third layer is called by scientists as the mesodermal layer. So this is mesoderm. Thus, during the embryonic development, we have three layers: an outer ectoderm, a middle mesoderm, and inner endoderm. Okay. So what happens now? So when the gastrula is formed, it is two-layered: outer ectoderm, inner endoderm, and there is a cavity inside the endoderm, and this is called by scientists as archenteron. Arc means primitive, enteron means gut. So this archenteron is referred to as primitive gut. Why? Because we are having an alimentary canal within which there is a cavity and there is a surrounding wall of the alimentary canal. According to them, the wall of the alimentary canal is derived from the endodermal lining. So, the endodermal lining gives rise to the wall of alimentary canal. Then what about the cavity, enteron? That gives rise to the cavity of the alimentary canal. Hence, we call this as primitive gut. Based on this observation, scientists say that endoderm develops into alimentary canal. That is its fate. So the fate of the cells of the endoderm is to develop into alimentary canal. But we know that alimentary canal starts with mouth and ends with anus. So coming to the gastrula again. So there is a gastrula where there is a cavity, archenteron, that opens to outside through a pore called as blastopore. So this blastopore should become either mouth or anus. So in some organisms, this becomes mouth. Then what happens? The embryonic development continues and this part becomes the anus. In such case, these are called as protostomians. Okay, where the blastopore becomes stoma means mouth. So proto means first of all. So these are the first kind of organisms where the blastopore in evolution becomes mouth. And after that is followed by the organisms which evolved to form where the blastopore becomes anus and this becomes on the other side mouth develops, based on which we call them as deuterostomians. Okay, so there are two types of organisms: protostomians where the blastopore becomes mouth and deuterostomians where the blastopore becomes anus, so that respectively on the other side anus and mouth are developed. In the meanwhile, what is happening? The endodermal, mesodermal cells go inside. This is one kind of development. In another kind of development, the mesoderm is originated along with the origin of coelom. Okay, and here there are three types of organisms based on coelom. Some are called as acoelomata, some are called as pseudocoelomata, some are called as eucoelomata. In acoelomates, there is no body cavity at all. In pseudocoelomates, there is a body cavity which is not surrounded on both sides by mesoderm, and it is nothing but a remnant of the embryonic blastocyst. Whereas in intracoelomates, the body cavity originates from mesoderm and it is surrounded on both sides by two mesodermal layers. The one which is present outside is called as parietal mesodermal layer. The one which is present inside is called as visceral mesodermal layers. And the cavity between these two, which is derived from mesoderm, is called as introcoelom. Sorry, eucoelom. Sorry, it is called as eucoelom. And the mode of development of this eucoelom is again of two types. One is called as schizocoelom, the other is called as enterocoelom. So, what is the difference between these two? So, let's come back again to the gastrula stage. In gastrula stage, what the scientists say here, the sorry, in the gastrula stage, the embryo, sorry, the endoderm forms into pouches which extend into the blastocele. This is the blastocele. This is archenteron. So here, what happened is, the endoderm forms into pouches, and this is archenteron. Hence, we call this entire thing as the roof of archenteron. Okay, the roof of archenteron is made up of endodermal layers, and from this small, what we call in imaginations arise, they go into the blastocele. Afterwards, what happens is they get separated. These get separated. How? They get separated and form into two pouches. So these are called as the pouches of archenteron. So the pouches of archenteron extend on both sides. They extend on both sides, enlarge in size until they meet each other. After they meet each other, what happens? Again coming back to archenteron, and the pouches extended, and where they meet they fuse with each other and leaving a space in the center. And we know that this is ectoderm, this is endoderm, and the newly formed layer is said to be the mesoderm, with a layer that is present towards the ectoderm called as somatic ectoderm or outer coelomic epithelium or parietal mesoderm or outer mesoderm, and a layer which is present towards the endoderm, which is called as splanchnic mesoderm, splanchnic mesoderm or inner coelomic epithelium or visceral mesoderm or inner mesodermal layer. Once there are two mesodermal layers encircling a cavity that is called as a body cavity, and such a coelom which originates from mesoderm, which is flanked on either side by two mesodermal layers, that arises from the fusion of pouches of archenteron, is called as enterocoelous coelom. Why? This coelom, that means this cavity, is nothing but the cavity of the archenteron. Hence, it is the enterocoelous coelom. So, enterocoelous coelom is formed by the fusion of pouches of archenteron. Okay, and these are called as enterocoelomates, and it includes echinoderms, hemichordates, and chordates. And in another kind of development, let's come back again to gastrula. So there are two layers, ectoderm and endoderm. Between these two, there are mesodermal layers, and these mesodermal cells, they form into what we call the mesoderm. And some scientists say that in some of these cells, there appears a vacuole, and this vacuole increases in size, and and passes through the entire mesodermal layer, and thus this splits the mesoderm into two layers, leaving a space in the center. Thus, here the cavity is formed by the of the mesoderm, splitting of the mesoderm. In Greek, Shaitos means breaking down or splitting. Hence, this cavity that is formed, the coelom that is formed by the splitting of the mesoderm, is called as Shaijocoelar coelom. And this is one case of what we call eucoelom because it originated from mesoderm. It is having two layers of mesoderm on both sides, and it is formed by the splitting of mesoderm. Hence, we call it as Shaijocoelar coelom. And this is exemplified by Annelida, Arthropoda, and mollusca. Annelida, Arthropoda, and molluscans are Schizocoelomids. Thus, we come across with the development of what we call three germinal layers along with the body cavity. Now, let us see the fate of these germinal layers. So, the first one is ectoderm. The scientists say ectoderm is divided into two parts again. One is called as somatic ectoderm and the other is called as neurectoderm. Okay, and the somatic ectoderm gives rise to body wall, especially the epidermis of the body wall, epidermis of the skin, and neurectoderms gives rise to nervous system. And secondly, the sorry, what happened here? Sorry, just a second. Okay, so I pressed the wrong key. Sorry. So what happened here is we are discussing at what point we have stopped here.
The somatic ectoderm or neuroectoderm, sir.
Okay, we have finished ectoderm, the fate of ectoderm. Next, endoderm. So endoderm, we have seen that it gives rise to alimentary canal from which the respiratory tract also develops. And lastly, we come across with the mesoderm. So ectoderm gives rise to epidermis of body wall and nervous system. Endoderm gives rise to alimentary canal from which respiratory tract develops as an adjunct. And coming to the mesoderm, it develops all the visceral organs plus coelom. From this, all the visceral organs and also coelom develop. The visceral organs include, apart from these 1, 2, 3, 4, the skin has dermis. So, the dermis of the skin develops where the muscles are present. The muscular system, the excretory system, the circulatory system, the reproductive system, the skeletal system, and the connective tissue. That means all the visceral organs along with the body cavity, connective tissue, and the skeletal system develop from mesoderm. This is the fate of these three germinal layers, and questions are being asked from these. And in the next class, we'll deal with the process of gametogenesis, how the sperms and ova are formed, followed by fertilization, cleavage, and gastrulation. Okay. So with this, we'll conclude the session today, and tomorrow, same time, we'll meet again. Thank you.